Assalamualaikum and greetings everyone. My name is Wan Saida Sauda Al-Huri, a final year student in Bachelor of Science Biodiversity Management, and I am supervised by my lecturer, Madam Sharifah Faiza Said Muhammad. Welcome to my final year project presentation, to my panels and all the viewers. So today, I'm going to present my project on title Diversity of Termite in Universiti Teknologi Mara, Jengka Campus Pahang. This is the outline that I will present after this. So let's start with the first part, which is introduction. Termites, which are members of the order Isoptera, are a group of insects with similar front and back wings. They are cellulose-eating social insects that live in nests or self-built terrariums. Termites are widely known as pests that cause harm to human property, but they also maintain nutrients that are vital to the health of the forest. Next is the problem statement. There are 3 problems that encourage me to do this project. The first one is the species of termite that inhabit UiTM Jengka Campus Pahang, and then the second one is the species richness and diversity of termite at UiTM Jengka Campus Pahang, and the third one is the preferable habitat of termite in Universiti Teknologi Mara Jengka Campus Pahang. Then the significance of the study, which is to know and recognize the diversity of termite species. Perhaps by knowing the termite species, we can better manage this pest. Next is the objective of the study. The first one is to identify the termite species at Universiti Teknologi Mara Jengka Campus Pahang. Second is to determine the species richness and diversity of termites in Universiti Teknologi Mara Jengka Campus Pahang. And the last one is to identify the habitat preference of termites based on their nesting and feeding groups. Then we go to the second part, which is the literature review. So the first topic is about the historical background of termite. There are 2 items in these topics, which are morphology of termite and hierarchy of termite. For the morphology of termite, according to Krishna 2012, Isoptera comprises 3 distinct body parts, head, thorax, and abdomen. Morphological criteria of termite encompass the shape of the head and mandible, the quantity of antennal segment, and measurement of body components. Termite colonies are composed of various castes, including primary reproductive, king and queen, soldiers, workers, and immature individuals. And in fact, their castes may be distinguished based on their morphology structure. The primary reproductive king and queen originated from wing form called alates that were prepared to swarm, mate, and establish new colonies. They have strong pigmented bodies and big compound eyes. Moreover, the caste of workers and soldiers in numerous species has no wings and are often blind. Besides, it has been documented that soldiers of termite have enlarged mandibles and heavily sclerotized heads. Next, for the hierarchy of termite. All termite species undergo the same life cycle. The life cycle of termites start when they spread their wings to colonize new sources. The winged reproductive males and females will leave their colonies to mate. Once the winged alate have completed their mating, they will shed their wings and form a new colony. This insect will then rule as king or queen in their newly established colony. Upon completion of the queen's fertilization, the eggs will hatch into immature termites. The immature termites, also referred to as nymph, and have the potential to grow into workers, soldiers, or flying alates. Once the colony reaches maturity, it proceeds to generate alates once again, thus perpetuating the cycle. The next topic for this part is the species richness of termite. This topic is divided into 2 items, which are species richness in Malaysia and in Pahang, since this study was focused in Jengka Pahang. So we go to the first item, which is the species richness of termite in Malaysia. Peninsular Malaysia has been recorded to have about 180 species of termites from 42 genera, constituting a more diverse fauna. The richness of termite assemblages within a given locality is undeniably impacted by the surrounding environmental condition. Heterothermus, Coptothermus, Prorhinothermus, Parhinothermus, and Scedorhinothermus are some of the genera that are frequently found in Malaysian forests. Next is the species richness of termites in Pahang. A previous study conducted by Jalaluddin and al. 2018 at Oil Palm Plantation and Darompin, Pahang had recorded about 29 species of termites from family Rhinotermitidae and Termitidae. Among the species found, Nasutitherium species were observed to be the most prevalent. Then, the last topic in literature review is the habitat preference, which are divided into 2 parts, nesting groups and feeding groups. In terms of nesting groups, termite naturally build a nest to protect their colony member. The nesting groups of termite can be divided into 4 main nesters: wood, arboreal, hypogeal, and epigeal. Moreover, in terms of feeding groups, termites can be categorized according to their dietary habits, including the soil feeders, which predominantly feed on soil, the soil-wood interface feeders, which prefer to feast on a combination of soil and wood, the wood feeders, who derive their sustenance from wood, the litter foragers, who scavenge for food in the form of decaying plant matter, the epiphyte feeders, who subsist on epiphytes such as mosses and lichens, the grass feeders, who graze on grass and consume dung, and finally some other minor feeding groups. Now, let's move on to the third part, which is methodology. This research was conducted at UiTM Jengka campus Pahang, with 9 locations had been chosen to identify the diversity of termite species. The sampling process was done between April until May 2023. All the collected termites were preserved in 75% ethanols in vials. The identification of termite species is based on the external morphological characteristic of soldiers that were carried out using a stereo microscope with reference to the published keys and descriptions, Termite Web website, Rudolph website, and previous studies also been used to determine the species. There are 3 sampling methods that I have used in this study: transect method, quadrat method, and active searching. For the transect method, each transect was partitioned into 5 segments as shown in the figure and the transect method. Samples were collected after 30 minutes. Next, this study also was carried out using the quadrat method as shown in the figure under the quadrat method. Besides, active searching sampling also was used as a sampling method. It involved manually collecting termites by hand from random location within the study site. Furthermore, the data were then analyzed using Markgraf index to measure species richness and abundance of termite species, while Shannon-Weiner index and Simpson index was used to measure the diversity of termite, and all these indices were calculated using PASS software. Moreover, the habitat preference in terms of feeding and nesting groups of the identified termites were analyzed by observing their nesting site with reference to the previous study. Now, moving on to the next part, which is result and discussion. A total of 12 species of termite from 8 genera with 2 families and 4 subfamilies have been successfully identified in this study. Next, for the species richness and diversity, roughly Microceratomus crassus is considered to be the most prolific species on record, having a total of 208 collected individuals, and family Termitidae is the most abundant species in record compared to Rhinotermitidae. In fact, most of the previous study, for example from Arumugam et al., also recorded family Termitidae as the highest family that usually found in research, since family Termitidae was the largest termite family. Moreover, the abundance and species richness of termites significantly differed on each sampling site. The termites were abundant at UiTM Front-Gate Hill, as shown by the values of Markgraf index with 0.6098, while the less abundant termite species in UiTM Jengka are at Infra Science Teknologi area, Hockey Field, Wood Tech area, Rumah Tamu Pon, and Fast Track area, with Markgraf index value is 0. The higher Markgraf index value indicates greater species richness in the community, and lower Markgraf index indicates less abundant species at the location. Shannon-Weiner index also shows that the diversity of termite species is higher at UiTM Front-Gate Hill, with the value 1.272. A higher Shannon-Weiner index value indicates greater diversity. It is also the same for Simpson's diversity index, where the index shows that diversity of termite species is higher at the same place as Shannon-Weiner index, which is at UiTM Front-Gate Hill. It is proved by the index value of 0.6953. The higher the index value, the greater the species diversity, and vice versa. Moreover, the lower species diversity of termites in UiTM Jengka Pahang are at the similar location of Markgraf index, which is at Infra Science Teknologi area, Hockey Field, Wood Tech area, Rumah Tamu Pon, and Fast Track building area, with Shannon-Weiner index and Simpson index is 0. The difference in the diversity of species in a place depends on several factors, among which are the environmental factors. As for the UiTM Front-Gate Hill site, 3/5 of the area is covered with trees, which means this area were quite rich with vegetation and far from environmental disturbance such as human traffic routes. Next, we proceed with the habitat preference of termite in UiTM Jengka Pahang. In this study, termite groups were found to be dominated by wood feeders. The habitats of the sampling sites were found to possess a greater quantity of decay wood, which potentially contributing to the higher presence of wood-feeding termite. Meanwhile, the least collected species are fungus growers, followed by soil feeders and intermediate wood soil feeders. Fungus grower required constant humidity, stable nest temperature, and low level of carbon dioxide. All these factors influence the presence of fungus grower termite. Besides, soil-feeding termites are highly sensitive to the changes in their environment, and they prefer shaded areas. The presence of canopies with moderate coverage at the sampling site may lead to a decrease in the abundance of the soil-feeding group. For intermediate wood soil feeders, lack of suitable wood sources and unsuitable soil type may be a factor in the fewer species present, since they rely on wood and soil as their food. In terms of nesting groups, there were 4 types of termites nesting groups found in this study, which are wood, arboreal, hypogeal, and epigeal nester. Most termite species are found from arboreal, epigeal, and wood compared to hypogeal nester. The difference in the number of termites feeding and nesting groups in 9 locations at UiTM Jengka, Pahang may be due to environmental factors found in those locations, such as temperature, humidity, food availability, land use, and habitat modification. For the last part is conclusion and recommendation. Overall, I can conclude that there are 12 species of termites from 8 genera with 2 families and 4 subfamilies recorded in this study. Besides, among the 9 study sites at UiTM Jengka, Pahang, UiTM Front-Gate Hill has the greater species richness of termites. Meanwhile, the lower species richness are at Infra Science Technology area, Hockey Field, Wood Tech area, Rumah Tamu Pond, and Fast Track Building area. In addition, 4 types of termites feeding group were discovered in this study, consisting of wood feeders, intermediate soil wood feeders, fungus grower, and wood litter feeders. Wood-feeding termites were dominated the area around UiTM Jengka among the 9 study sites. In terms of nesting groups, most termite species are from arboreal, epigeal, and wood compared to hypogeal nester. Last but not least, more sampling sites should be added because more termite species could be found, and this study needs to improve the trap by adding baits to the trap to attract the presence of termite into the trap. In addition, parameters can also be used to identify the environment preferred by termite species in terms of pH, soil type, humidity, and type of tree preferred by termites. Lastly, the identification of termite species can be further enhanced through the consideration of body measurements such as head size, mandibles, and head color. This is the list of references that I have used during my research. So that's all from me. Thank you for watching.